Hello and welcome to our Odin Analysis Spotlight video series. Today, we are going to take a look at how you can leverage the Odin Community Analysis software package to learn more about microbial communities like those found in soil using functional guild analysis and EcoPlates.
EcoPlates, containing 31 carbon substrates, are a powerful tool for carbon source preference screening for microbial isolates in communities. EcoPlates are a great screening platform for carbon source utilization as they're able to accept a wide range of sample types such as pond water, soil suspensions, purified microbes, and more. The EcoPlate contains 31 pre-plated substrates listed here, which were selected to probe a variety of pathways known to be important to environmental samples. While individual substrate utilization patterns are informative, it can be equally important to assess trends among substrates of the same type, otherwise known as functional guilds. Carbon sources in an EcoPlate can be divided into 5 functional guilds: carbohydrates, amino acids, carboxylic acids, polymers, and amines. Substrates within each of these groups required shared mechanisms or pathways to process, so it can be useful to compare the guilds to one another to more deeply understand the metabolic profile of an isolate or community. Here's an example of the type of comparison that's possible with functional guild analysis. Without the Odin software, you would need to export raw read data, manually annotate and compile reads for each substrate type, and generate this comparison. However, with the Community Analysis Toolkit, it's a simple and streamlined process that can be done directly in the software to generate publication-ready figures. So let's take a look now at the Odin software to more learn about functional guild analysis and how it can help shed light on differences between communities. Here we're going to be taking a look at 2 microbial communities in different soil samples. So first I'm going to select the project where my data is stored, and then I'll pick a reference sample. In this case we're going to look at CM1, which comes from soil rich in decaying organic matter. The test sample I'm going to compare to is CM2, and this comes from dry compacted soil with little organic matter. We'll then pick out our plate type, and then select area under the curve for our parameter, and then hit generate charts to get things going. Taking a look at the average area under the curve, or total energy output over time for each functional guild, it's really clear to see that the community CM1, shown here in orange, has a much more robust metabolic network, generating significantly more energy overall compared to CM2. This makes sense given that CM1 should be a more diverse and active microbial community, as it came from soil with lots of actively decaying organic matter. Additionally, we can see that while CM1 and CM2 are nearly evenly matched in terms of energy produced with carbohydrates, the differences are more evident when it comes to processing of more complex substrates like amino acids, polymers, carboxylic acids, and amines. This indicates that there may be a lack of microbes in the community for CM2 which are able to break down those substrates, or that the pathways required to process those substrates are absent or being turned off. I hope this demonstrates that functional guild analysis is a powerful tool for drawing impactful conclusions about the metabolic health and flexibility of your communities and isolates.
Thank you very much for tuning in to this Odin Analysis Feature Spotlight. If you have any questions or are interested in trying this analysis yourself, please reach out to info@biolog.com. Or to find more Spotlight videos and other helpful information, please visit biolog.com.